Welcome to the 21st Walker L. Cisler Lecture. My name is Hsiao-Ping Moore. I'm the Dean of the College of Arts and Sciences here at Lawrence Technological University. The Walker L. Cisler Lecture Series was established back in 1995 with a generous donation from the Holley Foundation. Mr. Cisler was the CEO and president of Detroit Edison, and he was also the founding member of the National Academy of Engineering. Besides being a noted industrialist, Mr. Cisler was known for his passion for science education. The Cisler Lecture Series is therefore created to honor his vision and to bring luminaries to come to our campus to help us promote science education throughout the Metro Detroit area. Before we introduce the speaker, I would like to invite our president, Dr. Verinder Madgil, to come up here and say his welcome.
Thank you, Dr. Moore. Good evening. Ladies and gentlemen, this is a very special occasion for Lawrence Technological University because not only is 21st Memorial Lecture, Cisler Lecture, we also have a very special guest who is admired intellectually, academic, physician, and a great leader of the first or the #1 public university in the United States. This is an extreme pleasure for us to welcome you and welcome him especially for his taking time. Remember, Michigan is playing tonight, but he elected to be here. That is called commitment. So I want to thank you all because you also have personal appointments and things to do, and you chose to honor Dr. Schlissel by your presence to listen to him and his views, at the same time to have kindness about Lawrence Tech to be here so you learn more about this university, which has a great beginning from 1932 during Depression, when Henry and Edsel Ford had a vision to create a different kind of university. And they thought about our founding president, Dr. Lawrence, who started this university, and we moved here in 1955 to this state, this particular site. During the Cisler Lecture, we had the great honor of having speakers of great eminence. They were thought leaders in science, technology, in social, political areas that touch science. And we had somebody from cybersecurity, which is a great issue that we are facing today. So any sphere of life that had intellectual component in science and technology, we had the very best come here. And today we have other great honor to have the President of University of Michigan, Dr. Schlissel, who in his own right is a noted scientist, a great leader of one of the great universities in the world and certainly in the United States. So one has to think about the people who came here, including Nobel laureates, thought leaders, societal leaders, and Dr. Schlissel belong in that group. It is so much an honor for Lawrence Tech to have him take time and be with us so that he can share his intellectual component of his career as a scientist, as a researcher, which is a great gift to the state of Michigan to have president of a great university who has that type of academic background. With that, please enjoy, and thank you again for being here. And I am just honored to have the chance to welcome you on behalf of everyone, and particularly our president visitor. Thank you.
Well, tonight we have a very, very special treat. Our speaker, Dr. Mark Schlissel, is a dear colleague of mine when we were both at the University of California in the Department of Molecular and Cell Biology. Mark is not only an outstanding scientist, but he has a big heart. I remember the first seminar lecture that we were teaching together, a student fell sick, and Dr. Schlissel jumped up, took off his jacket, and started performing CPR on him. So he always have students as first. So we're very, very honored to have him, and I'm honored to have been a colleague of his. Thank you. Now I'd like to introduce our provost, Dr. Maria Vaz, who is going to come up here and do the formal introduction.
Thank you very much, Hsiao-Ping. Dr. Mark Schlissel is the 14th president of the University of Michigan, where he started his mandate in 2014. In addition, he's a professor of microbiology and immunology, as well as a professor of internal medicine in the University of Michigan Health System. Dr. Schlissel received the Bachelor of Arts in Biomedical Sciences from Princeton University and the MD and PhD in Physiological Chemistry from Johns Hopkins University School of Medicine. He did his MD residency at the Johns Hopkins hospitals and the postdoc with David Baltimore, that, as you know, is a Nobel Prize laureate, at the Whitehead Institute at MIT. Dr. Schlissel became a faculty member at Johns Hopkins School of Medicine in 1991. In 1999, he moved to the University of California, Berkeley, as a faculty member in the Department of Molecular and Cell Biology, and in 2008, he became the Dean of Biological Sciences in the College of Letters and Sciences, and the C.H. Lee Chair of Biochemistry at UC Berkeley. In 2011, Dr. Schlissel moved to Brown University as the provost, and in 2014 became the 14th president at the University of Michigan. Dr. Schlissel research is focused on the developmental biology of B lymphocytes, the cell type in the immune system that secretes antibodies. His work has contributed to a detailed understanding of genetic factors involved in the production of antibodies and how mistakes in that process can lead to leukemia and lymphoma. He is the author or co-author of over 100 scientific papers and has trained 21 successful doctoral candidates in his lab. Nationally, he has served as member of the chair of the Immunobiology Studies section of the National Institutes of Health and on the Howard Hughes Medical Institute Scientific Review Board. President Schlissel was elected to the American Society of Clinical Investigators in 1998 and to the American Association of Physicians in 2013. He has been a member of the American Association of Immunologists since 1992 and was named a Fellow of the American Association for the Advancement of Science in 2013. He has helped organize major international scientific meetings and is a frequent seminar speaker at universities throughout the United States. Please help me to welcome Dr. Mark Schlissel to the 21st Annual Cisler Lecture. Thank you very much.
So hi, everybody. Thank you so much for the kind words of welcome, the very nice and thorough professional introduction, the nice memories, Hsiao-Ping. I caution you all, though, probably the last time that I resuscitated somebody was the time Hsiao-Ping mentioned. And that had to be perhaps 15 years ago, so I caution all of you, please don't, So what I want to talk about today is the role of curiosity and discovery, undirected research, discovery research, basic research it's often called, that ultimately is absolutely critical for advancing human society in very practical ways. We're in an era now, and I'll talk a little bit more about that at the very end of my talk, where people are questioning whether we're wise in investing in research, investing in discovery. I hope to give you a sense during this talk of how research just driven by curiosity, just by wanting to understand how something works, ultimately drives our economy and improves our health and quality of life. It spawns new industries, it employs hundreds of thousands or millions of people, and it touches every one of us through our healthcare system. Now, those of you that are hardcore real biologists will think this stuff is very simplistic that I'm talking about. And those of you that haven't studied biology since college will find this very obscure. So I'm going to try to hit the middle somewhere. The message isn't really the details of the science I'm talking about. It's how human curiosity, without limitation, without telling somebody what they must study or imposing a test of practicality on what they study, is the critical component to the major, major advances that completely change the world. So that's the take-home message I hope to get to. So, to me as a biologist, this picture represents the most profound thing that I ever learned. If you look at these things, in panel A, it's a bunch of bacteria, single-celled organisms. B obviously is a butterfly, C is a flower. There's a dolphin and a little baby, a child. If you just plop down on Earth from outer space, there's absolutely no reason why you'd think that at a molecular and cellular level, these things were almost identical. They look completely different. They behave completely differently. Yet, they're all made up of cells. They all use DNA and RNA to transmit and to store genetic information. They all use metabolic pathways, sort of biochemical reactions to carry out the processes of life that are so close to being the same, it's just remarkable. And then finally, they all go through various aspects of development where they sort of change fate through time. I'll talk a little bit about all those things. But the fact that there's so much similarity between all these different forms of life turned out to be critical for biologists to understand, not just the most, basic form of life, a bacteria, we'll talk a little about viruses, but also perhaps the most complex form of life as well. So what I want to talk to you about is the commonalities amongst all living things and how that's key to biomedical research. How discovery science, just driven by human curiosity, has been responsible for most of the practical, important advances, certainly in my lifetime. That basic science tools, the tools we develop as a consequence of discovery research, turn out to be enormously useful for things like diagnosis and the production of drugs and other therapies. And then finally, these basic discoveries require lots of work, something that's known in many places as translational research, translating these discoveries into forms that are actually useful for practical reason. So just to start with a little bit of levity, along with antimatter and dark matter, we've recently discovered the existence of doesn't matter, which appears to have no effect on the universe whatsoever. Now, this book has recently been reprinted by the Princeton University Press. It's by a famous scholar of education from the last century, Abraham Flexner, who had an impact on how medical education worked. And he also became the first director of Princeton's Institute for Advanced Study, a very advanced academic think tank, skewed towards the physical sciences, but not exclusively. And he pointed out with great clarity, looking back through history, how discoveries that at the time they were made, nobody knew, well, what is this useful? Why would we pay somebody to study this? And how the results ultimately become profound. So I just wanted to show you a copy of his little essay. And the different sort of types of science, one way to think about it, is on one end of the spectrum you have discovery science, where people are just following their curiosity. They just want to see how stuff works. There are basic scientists that try to get down to the molecular details of how a biological process works in the case of what we're talking about today. There's translational research that takes the insights learned about the very basic biology of life and tries to convert it into practically important things and medical implications. Then, of course, there's clinical research where we test new diagnostic modalities and treatments on patients. And then there's population health, how we try to take care of large communities of people based on the insights we've learned and developed initially from curiosity, but then all the way through to practicality. So that's the grand scope of the research I want to talk about. So this is an example from the early part of the 1900s, 1928, close to the founding of the university here. Very famous work that led to the discovery that DNA was the genetic material. So this is an experiment where Griffith took 2 different strains of pneumococcus, a bacteria that causes pneumonia, bacterial pneumonia in us, and can also make mice sick. If he takes a strain that he called the rough strain and injects it into the mouse is fine. Its immune system can get rid of this rough strain, no problem. If, however, he takes a strain called the smooth strain and injects it into the mouse, this is supposed to be a dead mouse, belly up. Now, what was really interesting is if he took this smooth bacteria and killed them by exposing them to high temperature, like when you cook something, it kills the bacteria, you inject the dead bacteria. If they were alive, they kill the mouse, but they're dead bugs, they don't kill the mouse. But the really interesting result is if you mix the bugs that don't normally kill mice with the dead bugs, now it kills a mouse. So the inference from that is something was transferred from these dead bacteria to the live bacteria that made those bugs lethal to the mouse. So this is almost 100 years ago, right? So what was this? And the process of figuring that out, this sort of discovery research, very famous work, Avery, McCarty, and McLeod, 15 years after that initial experiment. And what they did is they took the dead but other normally lethal cells, killed them, treated these cells either with an enzyme that destroyed proteins, an enzyme that destroyed RNA, or an enzyme that destroyed DNA. Then they mixed it together with live cells that were from the rough strain that doesn't cause disease. And they showed that if you add DNAs, these are no longer lethal. But if you add RNAs or protease, they're lethal. So that allowed them to infer that it wasn't protein, it wasn't RNA, but it was DNA that had the instructions to transfer a new property to a different organism. And that was really the first discovery that showed that DNA was the genetic material. No practicality at the time, just trying to understand how the chemistry of life works. Now, speed ahead to the 1950s. This is a very famous kind of data called an X-ray diffraction experiment that was used by the famous scientists Watson and Crick, along with their collaborators, to discover what the structure was of DNA. So imagine DNA can transfer this property of turning bacteria lethal, but how did it do that? What was the molecule like? How did it carry information? None of that was known. Crick was a postdoc, Watson was a graduate student. There's a very famous story about a woman scientist, Rosalind Franklin, who actually had the data that Watson and Crick kind of borrowed, in quotes, and that ultimately provided a higher quality data than Watson and Crick had. Her mentor joined these guys in the Nobel Prize. She was left out. Very unfair. There's a very interesting play that was written about this called Photograph 51, which was her famous photographic piece of data. If you ever get a chance to see it, I recommend it. So the structure of DNA, and it's this very familiar twisted double helix. This is a model of it. And the interesting thing about it is it has 2 different strands, polymers that go in different directions, but a G is always opposite a C and a T is always opposite an A. And everyone who's had basic biology since the late 1950s or early 1960s, this is what we should remember from biology, the double-stranded nature of DNA and the complementarity of the 2 strands. And in their famous 1953 paper, they wrote, "It's not escaped our notice that the specific pairing we've postulated immediately suggests a possible copying mechanism for the genetic material." And a decade later, they won the Nobel Prize for this work, essentially for this paper. And this is another depiction of the 2 strands with a G opposite a C and an A opposite a T. The idea is the 2 strands get separated, then new building blocks, new nucleotides join and form new second strands that allow 1 molecule of DNA to become 2 molecules of DNA. And that was the fundamental insight that came from this A's and T's and G's and C's base pairing in the famous DNA structure. So a decade worth of work in the golden age of molecular biology, which is really well chronicled in a book that's very accessible to the public called The Eighth Day of Creation by an author, Freeman Dyson, a historian. DNA is the material that stores information. RNA is copied from DNA and used to transmit the information and serve as a template for the synthesis of protein. And this is known as the central dogma of molecular biology. What I'm leaving out is this is completely wrong. That's an exaggeration. There are exceptions to each of these steps which are really interesting that go beyond the scope of what I could talk about. But this has basically happened starting in the 1920s all the way through the 1960s. 40 years, no practical application at all. 40 years, fundamental principles that explain really heredity and the reading of genetic information for all those different forms of life, from bacteria all the way to us. Purely undirected discovery, curiosity-driven research. Another interesting bit of curiosity-driven research had to do with a bunch of different labs around the world that were studying a special kind of virus called a bacteriophage that infects bacteria. So this big thing here, this is an electron micrograph of a single bacterium, and these things here that look like balls attached to stalks is a kind of virus that infects bacteria. And it turned out that some strains of bacteria were resistant to infection by the virus, and others the virus would infect and the bacteria would blow up. So of course scientists became curious, what is it that protects some strains of bacteria so that they don't blow up, and other strains suffer a lethal infection by this bacteriophage? So the answer has to do with a system that was known as restriction modification. There are genes in the bacteria that are resistant to the virus that restrict the ability of the virus to infect the bacteria. So then, of course, people wanted to know, what were these genes and proteins that protected the bacteria from being infected by the virus? And they turned out to be very famous enzymes called restriction enzymes. Those of you, again, that have studied biology, restriction enzymes. And what would happen is the restriction enzyme would recognize a particular sequence of the DNA that was being injected into this bacteria from the virus, from the phage, and then chop it up. And when DNA gets chopped up, it gets degraded, and it can no longer code for bacteriophage proteins that would take over and kill the cell. So those are called restriction enzymes. Now, what's special about them is they recognize there are lots of different restriction enzymes, and they recognize different specific sequences, and they make a double-strand break in the DNA, often leaving what are called sticky ends that can then join back up again. This basic thing is responsible for the whole biotechnology industry. It's responsible for all of recombinant DNA work, recombinant proteins, our ability to clone DNA, to sequence DNA, all depends on this. The person that did the biochemistry on the enzyme, a guy named Werner Aber in Germany, and Ham Smith, who was a postdoc at Michigan, so I had to get in a Michigan thing. Dan Nathans was Ham Smith's collaborator. These guys worked at Johns Hopkins, and they were my medical school professors years and years ago. So I have an emotional link. Dan has passed away of leukemia. Ham is still living, and Michigan gave him an honorary degree about a year ago. But these guys got the Nobel Prize in 1978 for discovering restriction enzymes and showing that they can cut DNA at specific places, and others showed that the DNA could be joined back up again. Now, why is that important? So the story goes back to the discovery of a kind of DNA called plasmid DNA. So it turns out that bacteria can have sex with each other. So bacteria do something called bacterial conjugation, where one bacteria will actually transfer DNA to another bacteria. That's actually one of the mechanisms of antibiotic resistance, where a resistant bacterium can transfer a plasmid that I'll tell you about in a minute that transfers the gene that codes for penicillin resistance or tetracycline resistance, for example. So this is a bacteria. This long structure here is called a sex pilus, believe it or not. and this bacteria transfers some DNA to this bacteria. What gets transferred is very often something called a plasmid. And a plasmid is a small circular DNA molecule, usually only a few thousand bases long, whereas the main chromosome in a bacterium is several million bases long, big and complicated. This is small and manipulable. This man, Joshua Lederberg, was the person who discovered plasmids and worked out a lot of the genetics of transferring genetic traits between bacteria. And he won the Nobel Prize for this. Again, what the heck is that useful for? But he won the Nobel Prize in 1958. Here he's getting the National Medal of Science from the first President Bush. Now, where this became really important was two scientists, Herb Boyer at UC San Francisco and Stanley Cohn at Stanford, were the first to show that you could take a plasmid, cut it open with a restriction enzyme, and then splice into it any piece of DNA you want, then close the circle back up again and put it back into a bacteria. And what that allows you to do is to clone this exogenous piece of DNA, grow it in a bacteria, so that you start with one bacteria and you can end up with 1 billion bacteria. And then you have 1 billion copies of that fragment you were interested in studying. And that's recombinant DNA, and it spawned this whole biotechnology industry. You know, one example of this biotechnology industry is a former colleague of mine and Hsiao-Ping's, Randy Schekman, a cell biologist, who used plasmids in yeast to synthesize insulin in yeast. So rather than purify insulin from cow pancreas, messy and complicated in lots of ways, he was able to take the gene that coded for human insulin and discovered the secretory pathway, or a lot of the secretory pathway components in yeast that allowed a company to express insulin on a plasmid in yeast and get it secreted and turn it into a commercial product, human insulin produced by recombinant DNA. Very practical outcome. If a drug company decided they wanted to do this and wanted to do it on purpose, without all those previous experiments over the course of 50 years, discovering that DNA was the genetic material, that you can cut and paste DNA using restriction enzymes, you can clone it in plasmids, never would have happened. But then we can take something, we can translate that knowledge into something incredibly practical, and it's actually spawned a $100 billion industry, right? All discovery research dependent. Other scientists worked on how can we determine the sequence of the letters in a molecule of DNA? How can we sequence a gene? Very famous work was done by a Nobelist, Arthur Kornberg, who worked first at WashU in St. Louis and then at Stanford University for many years. He won the Nobel Prize for discovering an enzyme called DNA polymerase that could make a copy of a DNA molecule. And then Fred Sanger actually won 2 Nobel Prizes. His first one back in the 50s was for discovering how to determine the sequence of proteins. And then 20 years later, he just developed a new mechanism to sequence DNA. And that led to the whole Genome Project, right? How do you determine the sequence of the letters? And I won't go through the details of how they did this because it gets very technical. But that same technology led to the development of a reaction called the polymerase chain reaction, or PCR. Has anyone ever heard of PCR? Okay, excellent. So what that is a way to take one copy of a DNA molecule and by iteratively separating the strands and then synthesizing a copy of each strand, separate them again, synthesize a copy, so it goes 2, 4, 8, 16, 32, 64, 128, and before you know it, you've got millions of copies of what your target DNA was. And the very clever thing that led to Kary Mullis winning a Nobel Prize for this technique was every step in PCR, you have to separate the 2 strands in order to synthesize new copies of each strand, and that gets done by heating it up. But every time you heat it up, you would denature and destroy the enzyme you needed to copy the DNA. So a number of people had the clever insight, let's get the DNA polymerase from a bacteria, Thermus aquatus, that grows in geothermal vents in Yellowstone. So they grow at 80 degrees. So the enzyme, the polymerase, is stable at 80 degrees. So you don't have to keep adding fresh polymerase as you do a PCR experiment. And now PCR is used very pragmatically for the detection of pathogens in food, in water, in tissue samples, in spinal fluid, in blood, very quick tests that allow you to look for all different kinds of viruses and bacteria, for example. It can help accurately diagnose genetic disease. It can do forensics. Those of you that watch, what's this TV series about medical examiners that's so popular? CSI. There are like 10 different CSIs. They use PCR all the time. You can distinguish different types of cancer from one another, huge number of different applications, all the result of a scientist in the late 1950s discovering the enzyme that copies DNA, and then these people turning it into an assay called PCR. I'm going to shift gears to closer to the area that fascinated me while I was running my research lab, and that's perhaps the biggest mystery of life. A sperm is one single cell. An egg is one single cell. A sperm fertilizes an egg, makes an embryo, and out pops a baby. How on earth does this happen? To this day, I look at this with the amazement of a teenager. So how a set of genetic instructions in a fertilized egg can end up coding for all of the capabilities you need to make a liver and a spleen and bone and blood and brain and connective tissue and gut. It's mind-boggling. Again, the target of tremendous amounts of basic science discovery research. This is the animal I studied in graduate school. It's called Xenopus Leavis. It's also known as the South African clawed toad, about the size of my fist. So, big but not unwieldy. We used to grow them in bathtubs in the laboratory before regulatory agencies said we couldn't do that anymore. But the reason people study these frogs is they have really big eggs you can see with the naked eye practically, that are a couple of microns, a couple of 1.5 millimeters or so, 1.3 millimeters in diameter. So these are 3 just been fertilized Xenopus eggs, and they begin dividing. I'll show you a movie in a minute if technology helps me. So they'll divide every 15 minutes or so, 2, 4, 8, 16, etc. And over the course of 2 days in a dish, 2.5 days, you can watch them turn into a tadpole.
This timing is very precise. Egg cells that have been fertilized at the same time divide and develop in almost perfect synchrony.
We didn't have music in the lab. So now it's a ball of about 4,000 cells, a hollow ball. Now it's developing a spinal region. Yeah.
After a day or two, embryonic development is completed, and tadpoles hatch from the eggs.
How can you not become a biologist? The curiosity of trying to understand this is actually what drew me into science and a little bit away from medicine. And starting with that one cell that you could see with the naked eye, you've now got a swimming tadpole 2 days later. How does that happen? Where's the information come from? How does the cells know to divide, when to stop dividing, when to become different types of cells? So I know you've made an investment and are very proud of your new stem cell center. So in a way, the birth of stem cell biology came from very famous discovery science, curiosity-driven research by one of my scientific heroes, a man named John Gurdon, who worked at Cambridge University in the UK, and this is right around 1970 or so. What John wanted to know is, we know that an egg and a sperm have all the information necessary to make a frog. Do all the cells in that frog still have all that same information? Does a skin cell remember how to regrow an entire frog? So what John did is he grew skin cells from a frog in a laboratory dish, and then he took an egg from a Xenopus and got rid of its nucleus. So now that egg has no genetic information of its own, and he took the nuclei from a nucleus from a skin cell and injected it into the egg. So now he has an egg that had none of its own genetic information, but just the genetic information from a cell that knows how to be a skin cell, and only a skin cell at the time. And then he puts it in that dish, like the experiment we just saw, and quite often, a few days later, you get a swimming tadpole. So the profound conclusion from that is, at least in this experimental model, all the information necessary to code for an entire organism is still present in all of the differentiated cells of that organism. I'm oversimplifying a little. And that you can sort of reverse the process that led that nucleus to know that it should be a skin cell nucleus. You can reverse it so it can now eventually give rise to all the different types of cells it takes to make a tadpole. And here's John in his lab. The guy worked at the bench, ran a 3-person lab his whole career, just amazing. He's probably still working at the bench. So that led to this, amongst the things, led to this notion of stem cells. So here is a fertilized egg, and this example is mouse probably, becoming an 8-cell embryo, now becoming an early blastula, a ball of cells. And these cells here, called the inner cell mass, you can put them in culture in a laboratory dish, you can make modifications to them using genetic tricks, and then get them to differentiate in a test tube, in a culture dish, to become either nerve cells or cardiac muscle or blood. So these cells are known as pluripotent stem cells. So when we do stem cell research here upstairs in your beautiful new lab, what we're trying to understand is how a cell of this type can give rise to many different functional types of cells. So the problem here, though, and what made this very controversial for some time, is extending this to humans required the use of fertilized human embryos as the source of these pluripotent stem cells. And you could think of all the ethical concerns, all the political concerns about doing stem cell research on, in effect, fertilized but not ultimately to be viable, but sort of leftovers from in vitro fertilization experiments that were designed to be therapeutic for infertile women or infertile couples. So this guy, Shinya Yamanaka, won a Nobel Prize in 2012. He actually shared it with John Gurdon, I showed you in a minute, for discovering that you could take skin cells as well as many other types of cells and add to them certain factors that turn them into pluripotent stem cells. So just like John Gurdon took a skin cell, put it in an egg, and the egg was able to convert that skin cell into a set of instructions for all these different types of cells, Yamanaka was able to take skin cells, add some clone genes to those skin cells, make them pluripotent stem cells, and now those stem cells can form blood and various cells in the gut and muscle. And this got rid of a huge amount of the ethical concerns, right? Because nobody really gets concerned if you give consent and I ask you for a skin biopsy. It doesn't seem to be as big a deal as taking a fertilized human embryo and turning it into an experiment. Now, there are many practical applications of stem cells. Many therapies are being developed that involve the use of stem cells to repair human deficiencies. Here's some work being done in our psychiatry department by a colleague named Mel McGinnis. If you want to understand what might be the biochemical defects in nerve cells from the brains of patients with depression, what's deficient compared to normal control volunteers, you don't really want to go around doing lots of brain biopsies, right? Pretty invasive. But what if instead you can do a skin biopsy, turn those skin cells into stem cells using those Yamanaka factors I mentioned a second ago, differentiate those stem cells into brain cells in a culture dish, and then study how they behave in an experiment. So that allows you to model a disease. And in this instance, McGinnis is looking at sort of electrical conductivity differences and differences in the response to lithium, a drug used in the treatment of manic depressive illness, in stem cells that became brain cells where the original donor had a major depression as compared to controls. And he's working out biochemical differences between brain cells from depressed and normals using skin biopsies to do the experiment. And they've developed a whole biobank of literally hundreds of stem cell lines from different patients with depressive disease, and then hundreds of control lines, trying to get at the basic biology. So that's one of the big advantages of doing stem cell research. Now, as another example of how undirected, curiosity-driven research can lead to great discoveries, I'm going to show you a news broadcast from 2 election cycles ago, from 2008. This is Rachel Maddow, remember the CNBC correspondent. Let's see if this works.
Projects that really don't make a whole lot of sense.
And this is Sarah Palin, remember.
Sometimes these dollars, they go to projects having little or nothing to do with the public good. Things like fruit fly research in Paris, France. I kid you not.
Do you believe that? Do you believe those stupid scientists studying stupid fruit flies? Fruit flies! We should obviously take away...
She was saying we should obviously take away our money. All right, so this is what you do with fruit fly research. So this is a famous fruit fly mutation. Here's the normal fruit fly. This fruit fly, instead of having antenna, has feet coming out of their head where their antenna was. It's called antennapedia. It's a type of mutation called a homeotic mutation. That's a mutation that changes the structure of a part of the body during development. Big deal, fruit flies. So several scientists over the course of about a decade, these 3 in particular, he was a postdoc for her, and Ed Lewis was working independently at Caltech. Eric went on to work at Princeton, and Nusslein Volhard is in Germany, discovered and cloned the genes that code for this family of mutations that could change the identity of body parts. And those were called homeotic genes. There are a whole cluster of them, and this is a fruit fly embryo. Turns out that highly related genes also exist in mammals that are homologous to each of those fruit fly genes. So wow, that's getting interesting, kind of. Turns out that mutations in these mammalian genes that are homologous to the fruit fly genes cause skeletal abnormalities in humans. These are human birth defects that we're seeing with mutations in different homeotic genes. So this fruit fly research done 30 years ago and awarded the Nobel Prize that defined the genes that are required to develop a body plan in a fruit fly that Sarah Palin said was silly. Why should the government pay for that? Led to a basic understanding of the organization of human development and the identification of a lot of human disease genes. So she was wrong. All right. So another major success driven by those decades of curiosity-driven research was the Human Genome Project. Remember Watson and Crick discovering the structure of DNA? Here they are back in the 1950s. This is what 50 years does to people. Crick and Watson on the right. A decade-long international project driven at the NIH by Francis Collins, who had been at Michigan for many years but moved to NIH to head the Genome Research Institute. Francis initially became famous for identifying the cystic fibrosis gene and a number of other disease-associated genes. And he went on to head the NIH. He's still NIH director, continuing into the new administration. The other group that worked on this was a private company led by a man named Craig Venter. There's an interesting backstory to this. Being a private company, the science world was very concerned that if he got the human genome first before the public project did, he would patent all the genes, and then he'd be the evil genius who controlled all products made from human genes. It turned out that courts invalidated his efforts to patent. He was trying to patent genes without knowing their function, so that became less of a worry. It turns out that his approach, though, was far superior to the one that was done in this international consortium. So the Genome Project actually finished up using the approach developed in Ventnor's company, but occurring at the NIH and then in funded labs in America and all around the world. It actually cost about $100 million to sequence the human genome. Lots of money, $100 million. Today, sequencing your genome or my genome costs about $1,000. In the coming years, just like you carry an insurance card, you're probably going to carry a microchip embedded in that insurance card that has the sequence of your entire genome, so that your medical care can be tailored to your own genetic predispositions. William Osler was a very famous physician at Johns Hopkins around the turn of the 18 into 1900s, that century transition. And he said, if it were not for the great variability among individuals, medicine might as well be a science instead of an art. And my doctor colleagues would snicker at this because we all think we're scientific. But it turns out that we are so different from one another genetically that the same disease in any two of us is likely to play out a little differently. We're going to be sensitive to different drugs because we're genetically different individuals. So I don't know if any of you have ever had the experience where a doctor says, let's try this drug. If it doesn't work, we'll switch to a different one. The reason why is we have genetic differences that will influence either the metabolism of the drug or even the underlying mechanism of the disease. And one of the great potentials of the Human Genome Project is the ability to see that genetic predisposition in one individual at a time, recognize that just because we call something a disease doesn't mean it has the same mechanism in you and me, and we be able to treat the cause of the disease one individual at a time. High throughput sequencing will give us very precise ability to diagnose disease, and then it has big implications for prevention, diagnosis, and treatment. Universities, including Michigan, but all around the world, are making major investments in what's called personalized or precision medicine. That's using DNA sequencing and other high throughput techniques to individualize diagnosis and treatment to you based on your own genetic background. And again, all began with the discovery that DNA was a genetic material, restriction enzymes let us clone genes, Watson and Crick determining the sequence, et cetera, et cetera. So as a humorous interlude, God, the human genome, the code's been unraveled. Damn hackers, now we have to change the password. So the last thing I'll talk about is a very recent major breakthrough that was awarded the Discovery of the Year in Science back in 2015, just a year and a half or 2 years ago. And conceptually, it turns out to be really similar. Remember I showed you the discovery of restriction enzymes? That restriction enzymes were a defense mechanism to allow bacteria to be resistant to viruses that would infect bacteria. Well, workers, 7 or 8 years ago, perhaps a bit longer, a decade ago now, discovered another one of these defense mechanisms. And these are two researchers who independently discovered, when they were looking at the DNA sequence of a bunch of different kinds of bacteria, an unusual set of repeated DNA sequences. So you'd have these diamonds indicate the same exact 100 nucleotide long sequence, repeated lots of times, but separated by sequences that were unique. And they puzzled, what is this? What could this possibly be? How could it possibly be used? So cutting a long story short, what it turns out is these unique sequences were actually captured from infectious organisms and became part of a mechanism that allowed the bacteria of this different strain to specifically recognize and chop up DNA from another one of these invading species worth of bacteria. So I'll show you a little more about that. The system is known as CRISPR Cas9. You can read it in Time Magazine and the New York Times. There's a big patent fight to see who owns it. But apart from that, what happens is these repeated DNA sequences get copied into RNA, get assembled with a protein into a complex that will specifically find a DNA target and chop it up, just like a restriction enzyme chops it up. But what these investigators realized, Jennifer Doudna and Emmanuel Champentier, working initially independently and then collaboratively, that you could put any sequence you want into this section of the molecule, and thereby you can target any sequence in the human genome. So I probably won't explain this well enough for people to understand, but if the genome is 3 billion nucleotides long, and DNA can have any one of 4 bases, A, G, C, or T, at any position. It turns out that a piece of DNA about 18 or 20 nucleotides long is enough information to be completely unique in the whole genome. It's not going to happen at random, given the size of the genome and the number of different permutations of A's, G's, C's, and T's there. Don't worry about it. So they can design a molecule that will recognize any single location in the 3 billion nucleotide human genome and chop right there. Now, the utility of that, the usefulness of that, is shown here. When you make a break in double-stranded DNA, it often introduces a mutation. It's also a site where a foreign piece of DNA that's homologous in the region can pop into the site of that break. Or you can delete and destroy a piece of DNA. So it's almost like now you're a surgeon with a knife, and you could look in the human genome for a mutation that causes a disease, and you could pop it out and replace it with the right copy. It's like science fiction. Using a system accidentally discovered by curiosity-driven research of someone trying to understand, why is there this funny repeated sequence in these weird bacteria? How random is that, right? Now, this is predicted to be a multi-billion-dollar industry that has the capacity to correct cystic fibrosis, to correct sickle cell anemia. If you know the genetic defect, you can repair it using this technology. Almost done. So it can inactivate any one of the genes in the genome. It can correct a gene. You can add a gene to a cell. It's known as gene editing. It could alter the function of a gene. CRISPR Cas9. If any of you want to invest in companies, look for the companies using this stuff. Now, the problem that comes about is, let's say, you've got a disease of your liver, where every cell in your liver has the bad gene. You can't actually correct 100% of the cells in a multi-billion cell organ. But what you can do is make stem cells from that patient, correct the defect in a stem cell, and then use that stem cell to replace all the deficient and defective cells. So here's an example from the hematopoietic, from the blood system, where you can take a hematopoietic stem cell that can give rise to all the different types of cells in the blood, and you can grow this cell in a laboratory. You can use CRISPR and Cas9 to fix a mutation. So you can fix the mutation in the beta globin gene that causes sickle cell anemia. You could fix the mutation in the SCID gene that causes severe combined immunodeficiency, the bubble baby disease, bubble boy disease. You could fix it in the stem cell, then do a bone marrow transplant. Put that stem cell back into the sick person, and now these cells, since they're healthy and they've been corrected, will take over and now produce the normal protein, and you've permanently cured their disease. So this type of approach right now is being done in experimental animals, and people are exploring some of the ethical concerns. Now, what if you want to go to a family that's got some terrible inherited disease? Should we be changing egg cells? Should we be changing the inherited nature of an individual's genetic composition? So on a piece of paper, this can be done now, and it's being done in experimental animals. Here's a fertilized, probably a mouse in this example, but it could just as easily be human. Here's a tiny micropipette injecting the CRISPR Cas9 enzymes and a targeting sequence to correct a gene defect, and you can grow that into an embryo, pop it into a foster mom, and out you get a baby. And that baby, their germline is now normal. So you've corrected their genetic disease in all of their progeny for the rest of time. Ethically, people worry about this sort of a lot. When you're treating a patient, that patient can give you consent. They can think about the implications of the treatment. The treatment will die when the patient dies. Here, you're changing the genetics of a human in a way that'll be passed on forever. If we can change things like disease genes, I always wanted to play basketball like my team is playing right now. I wasn't talented enough. What if we cloned the Michael Jordan gene and gave it to me or gave it to my progeny? And, that's a joke, but it's essentially, what if we're on this slippery slope of helping improve, in quotes, the human race in an inheritable way by genetic manipulation? So the scientific community is taking a little bit of a pause to the extent that scientists can be controlled. And there have been a number of international conferences, this was one of them, with many societies that sponsor this kind of research, talking about what are the ethical issues we really need to deal with before we move forward full speed ahead. So it would be irresponsible to proceed with any clinical use of germline editing until there's a broad societal consensus about the appropriateness of the proposed application. I'm guessing this is already being violated somewhere around the world right now. So I'm going to end here, and basically, this is my campus. This is the University of Michigan. Many of you are alumni. What has me completely jazzed about leading this wonderful university is I told you how you can go from discovery to basic research to translational research to clinical research to population health. We do it all right here on this campus. We've got great fundamental research doing discovery work. We've got an outstanding academic medical center taking care of patients, but also learning how to take care of patients better. And we've got one of the top 5 schools of public health in the country. So we can do this whole spectrum. The challenge will be whether the government's still going to support it. So this is another one of my scientific mentors and heroes, Harold Varmus. And Harold won the Nobel Prize for discovering oncogenes, cancer genes, back in the 1980s. At the time, he was at UCSF, and he went on to be the director of the NIH during its golden age, right, when its budget doubled. During the Clinton administration, he convinced the Clintons to do something bold. Let's double the budget of the NIH. He subsequently did something very unusual when his term was over and he had worked back at UCSF for a number of years. He got recruited back to the NIH to be head of the National Cancer Institute, the largest of the NIH institutes, but he went from being the director to a number of years later being the leader of one of the subsidiaries. But an outstanding cancer researcher. He's now at Cornell Med School in New York City. In between, for a decade, he was the head of Memorial Sloan Kettering Cancer Center in New York City, just an enormous contributor. Yesterday, he wrote an op-ed in the New York Times in response to the administration's skinny budget. And this budget calls for an approximately 20% cut to the NIH budget. It calls for a 33% cut to the EPA budget. We don't even know what the cut will be into the NSF budget because it wasn't even mentioned. It calls for the zeroing out of the National Endowment for the Humanities and the National Endowment for the Arts, $0. So Harold wrote a very powerful op-ed that I recommend to you in yesterday's New York Times, but he got at something that has me even more concerned and as citizens might concern all of us. He said that between those who believe in, there's a fundamental divide right now in our country, between those who believe in evidence as a basis for life-altering and nation-defining decisions and those who adhere unflinchingly to dogma. So the challenge here is it's up to politicians to make decisions. We elect them. It's a representative democracy. We've been that for over 200 years. The thing is, we want our leaders to consider facts and knowledge, and then they could decide what to do with it, but to make decisions based on information and to continue to fund the research that will give us the information to spawn new industries, to improve medical treatment, to understand how to steward our environment. And that's what Harold is pointing out as a major risk of the current era. So I'll end there, and I'll thank you for the honor of allowing me to deliver this lectureship. Thanks very much.
Thank you for that inspiring seminar. We have time for a few questions, so we have microphones, 2 microphones. So anyone would like to ask a question?
I left you all speechless. Sure.
Dr. Schlissel, thank you for that very interesting talk. And I know very little about this, but I have read about the occurrence of the Fibonacci number sequence in a number of areas in nature, and I'm wondering your thoughts on how that impacts on what you were discussing, sir.
Gosh, you've caught an area of complete ignorance. I don't know. Forgive me. I wish I was more of a mathematician. Thanks for school colors.
Oh, yeah, my pleasure. Dr. Schlissel, thank you. Tell me again, remind us, you mentioned, for instance, Watson and Crick, 40 years, this information wasn't practical. I think, everyone had to study it in biology and, bio class. But what motivates people like Dr. Watson and Crick and many of these others that you're talking about? Do you think some of them have the foresight? 40 years from now, somebody's going to apply my double helix to heal patients, or what's going through the mind?
Yes, that's a really good
researcher?
question. I think it's happening at two levels. One is, I think you have hit the nail on the head. Is it's not foresight with specificity to say how their work will become useful, but the confidence that basically trying to understand nature will eventually prove useful. That's on one level. On the other level, science is a very selfish pursuit. It's a real turn on. So think of the excitement of being the first person who understands what DNA looks like, knowing that DNA was a genetic material. It's a real rush. It's the thrill of discovery. It's for people that are curious. There are some kids, you'll leave your kid alone for a while, and before you know it, they're taking apart your washing machine or something in the house. I had one of those. It's driven by human curiosity, and that's its own reward in many ways. The rush, and for me, what turned me from somebody who was sort of heading towards a career in clinical medicine to someone who spent most of my life in a research lab was the thrill of being able to do something with your hands that made data that you could argue about with other people and then understand how something as complex as a gene or a cell works. The inquisitiveness and the satisfaction from learning little bits about something very complicated, for some of us, it's a real turn on. Yeah.
I have two questions, and to follow up on what you just said, how do we inspire curiosity with kids today when we don't leave them away from a screen long enough to find a box that they can turn upside down and find the parts to?
Yeah.
And then my second question is to addressing what you said at the end of your talk, and that is, what do we do to inspire curiosity in our politicians? And are you unifying university presidents who can speak up and speak back to save NSF and NEA, et cetera?
So both really good questions. So when it comes to how do we teach young people and give them the opportunity to develop this inquisitiveness and curiosity when it seems like they're plugged in most of the time? Yeah, I think a big change that's happening in how we educate students since we were students is we no longer need to teach students lots of facts because you carry the world's knowledge around with you in your pocket, right? So, standing up here, I could probably answer any question. I could have looked up the Fibonacci sequence and its role in biology if I was quick thinking enough. So we don't need to teach them to memorize lots of stuff. So what we're spending more and more of our time doing is problem and experiment-based learning. So we're having them experience the excitement of defining and solving a problem as a vehicle to reinforce the knowledge that we're teaching them how to look up when they need it. You see, so we're changing it into a much more personal, interactive, problem-oriented educational process, whether it's in science or any other field. And we're not there yet, but that's the big idea of how we either use the fact that kids are plugged in for good or get them unplugged and have them solving problems. The much harder question is, what do we do as professors, as concerned citizens, to make sure that collectively we continue to support curiosity-driven research, practical research, and then use the best knowledge that we have to make the best decisions that we can as a society. See, I don't think we can really teach politicians. I think we have to teach our students citizenship. I don't think we do a great job, by and large. Certainly my university has done a great job, but we're so loath to require anything specific of all of our students. But getting our students to embrace the responsibility to discuss and debate ideas using the facts and the tools we give them, not just with one another, but with their families, their neighbors, their relatives, often their communities and their workplaces, and then to place an extremely high value on voting, not to minimize the impact of voting. And then I think politics changes, right? So the question about what university leaders are doing, as the leader of a public university, I can't be a political actor, right? I can't come out for or against a politician, but I think that I can speak in favor or opposition to policies that impact my mission. So things that affect our ability to do research, I'm perfectly comfortable criticizing. Things that affect our ability to educate students, to build a diverse campus, to provide opportunities to lower socioeconomic and first-generation students. I can speak out about all that stuff. I think it's part of my job. I can speak up about primary education because that's the pipeline that feeds into the university. And then I can speak up about the role of facts and research in making good decisions. I just can't speak about a politician or a political party or issues that really aren't close enough related to the mission of the university because I'm so much a visible spokesperson for the university. And we live in a state with people who have very diverse opinions, which is the way it should be, right? University presidents in our different professional organizations are getting together, we're lobbying, we're putting out position papers, we're working with our internal experts, trying to put them in positions to have influence. When the new administration took over, I personally reached out to people with Michigan connections and offered the services of the university to help them in any way we might be able to. Because, we have a number of connections with current leaders in our new government. So this is how we try to engage. Yeah. Maybe one last question, okay, Hsiao-Ping? Yeah.
Hi. I have a question about the CRISPR Cas9 technology. I was wondering how soon do you think the scientific community will be able to agree on an ethical code so that we'll be able to implement it to the public?
Yeah, so that's a really good thing. So if you think about it, who is really responsible and who has the authority in the world to agree on how to move forward and to enforce that ethical standard on individuals in every country on Earth? So this is stuff that it turns out doing it, you can do it here in your labs upstairs. It is not, you have to be a good molecular biologist, that's about it. When it gets to the reproductive part with humans, then it requires special expertise, but it's the same kind of expertise that's available in many fertility clinics all around the world. In vitro fertilization has been going on for decades, right? So I think it's a really challenging problem to figure out who gets to decide and then to enforce the decision. Because people can be renegades and do this anywhere. So I think it's an open question what's going to happen. Our federal funding agencies certainly can say we're not going to fund research that violates an ethical norm that a scientific panel might end up suggesting, but then the private sector can fund this stuff. It's not that expensive. So it's a great question, but I don't think there's really an answer. And even though this panel that I showed you a picture of and their conclusion was just a year and a half ago, it turns out that these experiments are happening now on human embryos. They're not being implanted with the idea of making humans out of these experiments, but they're being done in the laboratory dish to work out the techniques. So it's happening already. I kind of don't know how we would stop it. It's a little scary. Yeah.
Okay, thank you.
Yeah, thank you.
Mark, we have a little gift for you. Would you please come down?
Sure.
And Dr. Magill would like to acknowledge a few people in the audience.
Thank you. Dr. Schlissel, this was a historic lecture, and I hope you will all agree, right? And only who has reached that level of understanding can make us understand, and he did so very well.
Thank you very much.
Thank you very much.
Yeah.
And on behalf of the Lawrence Tech community, our friends, alumni, Michigan alumni, as well as LTU or others who took time to be here to listen to Dr. Schlissel, and particularly some folks who have guided this university over the year and continue to do so, I'd like to acknowledge with your help 3 people. Dr. Marburger, our 4th president. And our board of trustees who guide us every day when we have needs and we present to them and they thoughtfully help us to advance the institution. Our board of trustees, Trustee Lauren and David Wallin. And on behalf of you, this is a very token gift. What can we do to a University of Michigan president? We are not even capable. But this has our heart in it. Thank you very much.
Thank you. Thank you, that's really kind. So kind. Yeah. Oh, well, thank you very much. It's a real pleasure. Thank you.
We have a dessert reception out there, so please enjoy yourself.